Hello everyone, welcome to our channel Bio Ocean, and this is me Sheethal Maria with a new topic about the tissues. I hope that you all are very well known about the tissues, but still I want to give you a general idea of what tissues are, their classification, their peculiarities and all those stuffs. So let's get into the video. And before that, if any one of you are new to my channel, please subscribe to my channel and press the bell icon so that you can get the notifications whenever I upload a video. What are tissues? Let's discuss that first. Tissues can be defined as a group of cells. What are cells? Actually, cells are the basic structural unit of life. And here, these tissues are made up of a group of cells with a common origin, and usually they will be performing a common function. And every organism is made up of different kinds of tissues. And according to their capability of dividing or not dividing, these tissues can be classified into two main groups, that is, meristematic and permanent. So let's understand what meristematic tissues are first. Meristem are those restricted large regions in plants of growing plants where active cell division occurs. And that region in plants is defined as meristems. And this meristem is the word which was derived from a Greek word called meristos, which have a meaning divided. And plants have different kinds of meristems. The meristems which occur at the tip of roots and shoots and produce primary tissues are called as apical meristems. I have provided you a picture showing the meristems, apical meristems of root and shoot. There you can understand where those meristems develop in case of root as well as in case of the shoots. Okay. Here, the root apical meristem occupies the tip of the root, while the shoot apical meristem occupies the distantmost region of the stem axis. And during the formation of leaves and elongation of stem, some cells are left behind from the shoot apical meristem, and they are called as the axillary bud. These buds are present in the axils of leaves and are capable of forming a branch, otherwise a flower. And the meristem which occur between mature tissues are called as intercalary meristem. And they majorly occur in grass, but grazing animals usually consume these regenerated parts of these grasses. And both these apical and intercalary meristems together constitute to form the primary meristem because they appear early in the life of a plant and contribute to the formation of primary plant body. And the meristems that occur in the mature regions of roots and shoots of many plants, particularly that produces woody axis and appear later than primary meristems, are called as secondary, otherwise lateral meristems. They are cylindrical meristems. And following the divisions of cells in both primary as well as secondary meristems, the newly formed cells become structurally and functionally specialized and lose their ability to divide. And such cells are called as permanent, otherwise mature meristems, and they constitute the permanent tissue. During the formation of the primary plant body, specific regions of apical meristem produces dermal tissues, ground tissues and vascular tissues. Now let us understand what permanent tissues are. The cells of the permanent tissues usually are having no divisions further, and they will be in similar structure and function and together constitute to form simple tissues, while those permanent tissues which are having different types of cell are called as complex tissues. First of all, we will learn what simple tissues are. Okay, a simple tissue is made up of only one type of cell. And simple tissues are mainly parenchyma, collenchyma and sclerenchyma. These are the three main types of simple tissues. So let's understand what parenchyma are. Parenchyma forms the major component within the organs, and the cells of parenchyma are always isodiametric. They may be spherical, oval, round, polygonal, or elongated in shape. They have thin wall which is made up of cellulose, and they are either closely packed or have smaller intercellular spaces. The major functions of these parenchyma constitute photosynthesis, storage, and secretion. Then, the collenchyma occurs in between the layers below the epidermis in most of the dicotyledonous plants. It is either found as a homogeneous layer or in patches. It consists of cells which are thickened at the corners due to the deposition of cellulose, hemicellulose, and pectin. They can be oval, spherical, and polygonal in shape, often contained with chloroplast. And these cells assimilate food particles when they contain chloroplast. Intercellular spaces are absent here, and they provide mechanical support to the growing plant parts such as young stem and petiole of a leaf. And the sclerenchyma consist of long, narrow, thick and lignified cell walls having a few or numerous pits. They are usually dead and devoid of protoplast. On the basis of variation in form, structure, origin and development, sclerenchyma can may be either fibers or sclerites. And the fibers are thick-walled, elongated and pointed cells generally occurring in groups in various parts of the plant, while the sclerites are spherical, oval or cylindrical, highly thickened dead cells with very narrow cavities or lumen and are commonly found in the fruit walls of nuts, pulp of fruits like guava, sapota, pear, etc. Now let us discuss about the complex tissues. These complex tissues are made up of more than one type of cell and they work together as a unit. Xylem and phloem are the main constituents of these complex tissues. Xylem functions as a conducting tissue for water and minerals from the roots to the stem and leaves. It also provides mechanical strength to the plant parts, and majorly the xylem is composed of four different elements. The first element is tracheids, which are elongated or tube-like cells with thick and lignified walls and tapering ends. These are dead and without protoplasm. The inner layer of the cell walls have thickenings which vary in form. In flowering plants, tracheids and vessels are the main water transporting elements. And the second element is vessels, which is a long cylindrical tube-like structure made up of many cells called vessel members, each with lignified walls and a large central cavity. And the vessels can also be devoid of protoplasm, and the vessel members are interconnected through perforations in their common walls. Their presence of vessels is a characteristic feature of angiosperms, while gymnosperms lack vessels in their xylem. The next element is xylem fibers, which have highly thickened walls and obliterated central lumens. They may be either septate or aseptate. The fourth element is xylem parenchyma, which are the cells living with thin-walled cell walls made up of the cellulose, and they store food materials in the form of starch and fat and other substances like tannins. The radial conduction of water takes place by the reparenchymatous cells. The primary xylem is of two types, protoxylem and metaxylem. Protoxylem is the first formed xylem, while the later formed xylem elements are called metaxylem. In the stems, the protoxylem lies towards the center and metaxylem lies towards the periphery of the organ. And this type of primary xylems are called as endarch, while in the case of roots, this occurs vice versa. That is, the protoxylem lies towards periphery and metaxylem lies towards the center. In such an arrangement of primary xylem are called as exarch. Now let us discuss about the phloem. Phloem mainly transports food materials usually from the leaves to other part of the plants. Phloem in angiosperms is composed of sieve tube elements, companion cells, phloem parenchyma and phloem fibers. Now let us understand first what phloem parenchyma are. Phloem parenchyma is made up of elongated, tapering and cylindrical cells which have a dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pits throughout which plasmodesmatal connections exist in between. The phloem parenchyma stores food materials and other substances like resins, latex, and mucilage, and it is absent in most of the monocotyledons. And the next one is phloem fibers, which are bast fibers made up of sclerenchymatous cells, which are generally absent in primary phloem but are found in the secondary phloem. They are much elongated, unbranched, and have pointed needle-like apices. The cell wall is quite thick, and at maturity these fibers lost their protoplasm and become dead. The phloem fibers of jute, flax and hemp are commercially used. And the next one is sieve tube elements, which are also long tube-like structures arranged longitudinally and are associated with the companion cells. Their end walls are perforated in a sieve-like manner to form the sieve plates. A mature sieve element possesses a peripheral cytoplasm and a large vacuole but lacks a nucleus. The function of sieve tubes are controlled by the nucleus of companion cells. And the first formed primary phloem consists of narrow sieve tubes and is referred to as protophlloem. And the later formed phloem has bigger sieve tubes which is referred to as metaphloem. And the last element is companion cells, which are specialized parenchymatous cells which are closely associated with sieve tube elements, and the sieve tube elements along with companion cells are connected by pit fields present between their common longitudinal walls. The companion cells help in maintaining the pressure gradient in the sieve tubes. This is the general idea along with the characteristics of what the tissues are. Hope you will understand what I've said, and if you have any kind of doubts, let me know through my email ID. And also, if you like this video, please give me a thumbs up, comment your suggestions and share it to your friends and others who are interested to learn biology. Thank you for listening. Let's see you in the next video. Thank you. God bless.